Sir, you can continue.
Yes, ma'am. Thank you. Good evening, Ajay.
Good evening, sir.
Good evening, Kanam. Last class, what we have discussed general outline of the DNA, isn't it? Today, okay, today, so we are going to discuss about very deep aspects. So you know about the DNA as a double helical model. Okay, it has a two strand. This is the structure of double helical structure. Helical means twisted manner. Okay, it composed of more than one polynucleotide. Already we discussed. Nucleotide is nothing but three components is there. One is sugar molecule and a nitrogen base and phosphate also there. Okay. So this structure, the sugar phosphate act as a backbone because it is always occupying this area only. Okay. So the outer layer. The center there is a nitrogen basis with the which bond already you know about hydrogen bond, isn't it? Here the phosphate bond is outside that inside there is a hydrogen bond correspondingly, which one you know? Adenine, thymine, cytosine, and guanine, otherwise called as guanine and cytosine respectively, with a specific double bond and triple bond. Okay. Okay. See here. So DNA double helical model. So DNA is composed of two polynucleotide chains. Okay, the two polynucleotide chain means you know about 5'N, 3'N. Okay, it is anti-parallel with each other. So 5' and 3'. The opposite stand is also 3' and 5'. So two polynucleotide chains and large macromolecules. It is twisted anti-parallel chain held together by which bond is there? No, hydrogen bond. Why it is hydrogen bond? See that structure. This is the 5'N. Here it's end. Where there is a end, this is called as a 3' end. So here 5' means opposite side there is a 3' end. It is end with the 5' end. Okay. In between there is a nitrogen base pair with the hydrogen bond. Already we discussed. Okay. That is given here. See. And sugar and phosphate forms the backbone. Sugar and phosphate forms the backbone. Where is the backbone? So this is considered as a backbone. Suppose that sugar and phosphate, it is not present here. What happened? No, the nitrogen, they do not play a role. So the double helical structure is not possible. Okay. So the backbone is, so just like a insertion of the nitrogen base on the phosphate or sugar molecule, like that you have to imagine. Okay. So this is the entire structure of the thing. See here, sugar and phosphate forms the backbone. Nitrogen bases forms the interior pad through the hydrogen bond. Complementary base pair is an important feature of the DNA structure. Complementary base means, what is the meaning? Adenine always combined with the molecules of thymine. Guanine combined with the molecules of cytosine. So that is complementary each other. Suppose it will not join the adenine to guanine, so cytosine to thymine, like that it will not pair because it is a, what is a, it's not a correct matching. Okay, always this is a rule of, what is a Chargaff rules. Okay, Chargaff rules, what is that? You know, one adenine or one purine molecules join with the another pyrimidine. So adenine is a purine molecule, thymine is a pyrimidine molecules, guanine is a purine molecule, and cytosine is a thymidine molecule. So this is the normal pair. You can ask the question, sir, why the adenine it will not join with the cytosine? The Grigg rules already told you. Which one you know? One purine join with the pyrimidine only, no? Adenine, why it will not join with the cytosine? Guanine, why it will not join with the thymidine? Crassly. Okay. So that is the rules. Okay. Then only the structure will be maintained. That is very, very important. Okay. Idea. Okay. See here. So this is the structure. Already we know about. So the sugar molecule is here. Addition with the phosphate molecules. Okay. The stand. This is the stand. Okay. Double helical. This is the one stand. This is the another stand. In between, there is a nitrogen bases there. Okay. Here, starting from the 5' end and the end with the 3' end. This is the structure has given us a straight manner, but here it is a twist manner. Is that okay? See, 5', 3'. So anti-parallel means see here 5 and 3. Here 3 and 5. So 5', 3'. Here started 5'. Here end with the 3'. So these two, what is that? Backbone is composed of which one you know? Sugar and phosphate. The center there is a nitrogen base is there. Okay. The structure is twisted manner. Okay. Already we discussed, isn't it? So this is the structure. Each and every rotations, what happened? How many molecules present you know? 10 nitrogen bases. The 10 nitrogen. 1, 2, 3, 4, 5, 6, 7 only given here. So in between these, what is that? This groove. Okay. To this groove, 10 molecules present in it. In between there is a 10 molecule. Each one it has a 3.4 Armstrong. 10 into 3.4, you will get what is that? 34 Armstrong. See that 34 Armstrong is. So this condition is called as this is the major groove. This is the minor grooves. Small one, this is a large one. Like that is also divided. The in between the length. So diameter. Diameter means from this portion to this portion. This portion to this portion. Okay. 20 nanometer, otherwise called as 20 Armstrong. Don't forget, my dear students. DNA diameter 20 Armstrong. Each and every groove there is a 10 pair of nitrogen bases. Okay. In between the one nitrogen base as well the another nitrogen base, there is a space is there. That space is called as 3.4 Armstrong. 3.4 Armstrong. 10 into 3.4, 34 Armstrong totally. Okay. Got it? Any doubt in this area? Okay. Go ahead.
Sir, no doubt, sir.
Okay. What about Dhanalakshmi?
No doubt, sir.
Okay, go ahead. See that. The two polynucleotide chains have an anti-parallel polarity. 5', 3', 3', 5'. Okay. The two chains are coiled in a right-handed fashions. Okay. When you are going to study the different types of DNA molecules, right-handed, left-handed. In sugar molecules also, in amino acids also, it has a specific rotations. Levo-rotatory, dextro-rotatory, like that they told. Okay. D-glucose molecule sometimes they give. L-glucose, D denoted as dextro. L denoted as levo. Likewise, the DNA structure also, it has a two side only right side or left side. That is also right-handed fashion or right-handed helix. Suppose if it is left-handed fashion, left-handed helix. Which one is the left-handed? You know, just imagine there are 4 types of DNA. 4 types of DNA present in it. A, B, C, and jet. The jet DNA always in left-handed rotatory. Otherwise called as left-handed helix. We will see. Okay. So don't forget, my dear. Uniform distance is maintained between the 2 strands of helix. This is also very, very 10 pair, 10 pair, 10 pair. It may be varied, depends upon the types of DNA molecules. Suppose we see A or B or C. Now we are studying the form is B form, right-handed helix. This was discovered by Watson and Crick. Okay. See here. Each strand has a 10 base pair, 34 Armstrong. 34 Armstrong. Don't forget. How many pairs? 10 pairs. Already told you, isn't it? See that. And diameter of helix also 20 Armstrong. That things also already I told. Okay. Not only that, and also some of the other organs, such as which one, mitochondria as well as the chloroplast. Not only in the nucleus, the DNA is not found in the nucleus only. Sometimes other than the part means nucleus, apart from the nucleus, some of the components, some of the cell bounds organelles also present DNA molecule. They are called extranuclear. See here, the DNA found both nucleus and cytoplasm. Nucleus already we know, isn't it? Cytoplasm, what are the components, The extranuclear found in the mitochondrial and chloroplast. See that this is the mitochondrial structure. Okay, this is the cross section, this is the off. When you are going to take the cross section, inside there is a nucleus is there. Okay, so the this is the cell structure. Sorry, this is the cell structure. The mitochondria is isolated here. The mitochondria inside, what happened? There is a circular DNA present. This DNA is called as mitochondrial DNA. This condition is called as, what is that? Extranuclear inheritance. This is also a extranuclear. Extranuclear. Nuclear means extra from the nucleus. Okay. Other than the nucleus, additional DNA molecules also present outside the nucleus in the cytoplasm of the cell, particularly mitochondria. Mitochondria, it has a, the extra chromosomal body, that is called as mitochondrial DNA. Likewise, the chloroplast also it has. What is that? See that? So this is the mitochondrial structure. See that cell structure? So inner side there is a mitochondria. This is a mitochondrial structure, already know. So the finger-like projection is called as, what is that? Cristae. There is a F1 particle is there. The respiration also takes place. Adenosine triphosphate formation. That is called as high energy compound. Otherwise called as energy currency. Okay. That thing's also already know, isn't it? Here, the structure of the mitochondrial DNA look like this one. This is called as a circular manner. At the same time, helical structure is there. Okay. Are you clear? Okay. So go to the next one. Okay. So this is the chloroplast. So the chloroplast is also here. They have told, see here in this part, nucleoid. There is a DNA ring is there. This portion is called as nucleoid. This is the mitochondria. Already we saw it, isn't it? So likewise, this is also present in the outside the cell, isn't it? It is not present inside the nucleus. It is outside the nucleus. That is called a cytoplasm. So this is also present in the plant cell. This is the animal cell. Animal cells also, what is that? Mitochondria present. But in the case of plant cell, chloroplast as well as mitochondria present in it. Both are present. But animal cell only, in the mitochondria only present in, particularly the euglena, it possesses which one, chloroplast also, the animal. So it is a controversy effect. Okay. Just here, you just note it down, which one, the animal cell is also mitochondria, it has an extra chromosomal body. Likewise, plant cell, particularly the chloroplast, it contains which one, extra chromosomal body. They possess their specific characteristics because the presence of nucleotide. Okay. So go to the next one. The packaging of DNA helix. This is very, very important, my dear students. Packaging of DNA. How it will be packed? What are the components? What are the distance? What are the length of the DNA? How many nucleotide present in it? That things you have to note. And take a notes, my dear student. This is very, very, very important one. Okay, see here. The length of the DNA is found to be far greater than dimension of the typical nucleus. Okay. So the length of the DNA is found to be a far greater than dimension of the typical nucleus, particularly the total number of base pair in a typical mammalian cells, that is equal to, note it down, my dear, mammalian cells, the base pair equal to 6.6 into 10 to the power of 9. Here you have to put the 9 in the upper portion. Okay. 6.6 into 10 to the power of 9. Take a notes, my dear. And distance between two base pair already we discussed. What is that? 0.34 nanometer. Nanometer 10 to the power of -9, isn't it? Okay. Therefore, length of the DNA, how it will be? You have to multiply these two. Okay. You have to multiply these two. You will get the length of the DNA. So 0.34, this one, into, which one you know? 0.34 into 10 to the power of -9 into 6.6 into 10 to the power of 9. That is equal to 2.2 meter. So normally the human, what is the length of the genes, DNA molecules? So 2.2 meter. This is also very, very important in the NEET question. Particularly, they ask the question in human being, the length of the DNA molecules, it will be, which one of the following is a correct one? Like that they may ask. 2 meter, 2.5 meter, and 2.2 meter, 2.8 or something else. Okay. How will you calculate? Just imagine, this is the normal one. Okay. 2 base pair in between the distance like this. I have to multiply. The mammalian cell contain how many numbers of pairs? So multiply, you will get this much. Got it? Okay. So go to the next one. Note it down, my dear. Note it down, my dear students. Please take a notes kindly. Okay. See that. The size of the nucleus is of the order of the 10 power -6 meter. Size, it may be vary. So length of the DNA is greater than the size of the nucleus. The length of the DNA is greater than the size of the nucleus. Size will be very less in nature. What is that? How many Armstrong is there? In between 20 Armstrong only, that in between the size, but the length will be very large in it, 2.2 meter. Okay. Okay. Go to the next one. This is also very, very important one. Some of the bacteriophage also having the nucleic acid, isn't it? The bacteriophages 174, they have given us a name, lambda 174 here, the lambda. Symbol is Y is a inverted manner. Already you know what the symbol. In physics already you studied, isn't it? The bacteriophage 174 has 5,386 nucleotide. Note it down. Sometimes they ask the question, match the following. 5386. Just imagine 5 plus 3, 8. Okay, after 5, 6. You have to multiply like this. So you have to calculate like this. You can mnemonics like this. Imagine or remain like this. Okay, 5 plus 3, 8. So after 5, 6. 5386. And bacteriophage lambda. See here. So this one is 48,502 base per percent. This is also asked sometimes. This is also one type of the bacteriophage. This is another type of bacteriophage is also there. And E. coli has, this is very, very important. This is a bacteria, isn't it? Bacillus type. 4.6 into 10 to the power of 6 base pairs. Note it down. This is also very, very important. Okay. E. coli has a 4.6 into 10 to the power of 6 base pair. Next. See, this is the DNA structure. This is a single strand already in our board. This is a double stranded structures. In between, there is a nitrogen. This is RNA. This is the DNA. Okay. RNA has a single strand. DNA has a double strand. These are the nitrogen bases. Which one is different? No? Uracil is different. But here, thymine is different. Thymine present only in the DNA. But instead of thymine, here RNA, uracil is present. Okay. Don't forget, my dear. Okay. So, DNA is an acidic substances in the nucleus. Was first identified by Frederick Meisser. Already you know relevant. In the historical aspect, already we studied. 1869. Note it down, my dear. Year is very, very important. Sometimes they may ask question year also. 1869. He names it as nuclein. Okay. From the pus cell. Okay. From the pus cell. Not only that, and also it contains DNA as well as the protein molecules. Already we studied in the historical aspects, isn't it? This is very, very important. Next one, you have to put 1,000 times is very, very important because it is human genome. It is a haploid. Human genome. Take it down, my dear students. Ajay and Dhanalakshmi, take this one, is very, very important. human genomes contain 3.3 into 10 to the power of 9 base pair. You have to put like this. 3.3 into 10 to the power of 9 base pair in a human genome. Because they asked the question, because we are all very important. Okay, particularly human genome, lambda phage, E. coli. Question will be there in the NEET. 1953, double helical structure already in our lab. DNA was given by James Watson and Francis Crick based on their X-ray diffraction. Yesterday, Ajay asked, what is meant by X-ray diffractions? Isn't it? Already I told you, it is related to X-ray, okay, methodology, and also X-ray crystallography. Crystal means any one of the solid material. When we are passing X-ray, what happened? The image will be fall onto the opposite side or film, okay, just like a X-ray. This was discovered by which one? X-ray diffraction data produced Maurice Wilkins and Rosalind Franklin. Without these diffractions, the X-ray diffraction, the James Watson and Francis Crick, it will not give a structure of give you a structure of which one? DNA molecules. This is also very, very important. Without this information, James they will not produce the DNA structures. That is very, very important one. See that X-ray diffractions. X-ray diffraction is a method of X-ray crystallography, in which a beam of X-rays strike a sample. That's why I told you crystalline substance or solid substance. So DNA molecules also converted into solid substances land on a piece of film or other detector to produce a scattered beams, just like a film. Okay, so in between the film, the beam is there as well as the screen is also there. When a light pass onto the film, what happened? The film is what the picture they are having. It may be fall onto the screen, isn't it? Likewise, see that next we will see these beams make a diffraction pattern of the spots. The strength and the angle of these beams are recorded as a sample is gradually rotated. Okay, see the next one, you can easily identify. This is the structure. So here they have given as a DNA crystallized form. This is the X-ray beam is there. After passing the X-ray through the DNA model, you will get the diffraction pattern, different diffract portion. So we can easily calculate which one is a high concentration, whether adenine, guanine, cytosine. Likewise, we can easily identify it. How it will be the result? How it will be the result? See that this is the way of result. See that this is the structure of after getting the X-ray diffractions. Started from the 0 and 5 and 10. See the helical X. Helical means rotation will be like this. See, so this is a 0 and 5 and 10. Okay, as a result, you will get the what is the result. This is the methodology that were used for the identification of the solid particles. Solid particles, just like a DNA, RNA, protein structure, three-dimensional structures, and other components also we can easily identify. Okay, so that's all about it. Are you clear now, Ajay?
Yes, sir.
Okay, good. So go to the next one. So in human genomes, there are so many types have been reported. The human genome also broadly divided into two types. One is called as nuclear genome and mitochondrial genomes. Okay, how many base pair is that? 3000 MP megabytes. 30,000 genes also there. Okay, the 30,000 nuclear genome is also divided into genes and gene-related sequences. The extragenic DNA. So 70% of extragenic and 30% of gene and gene-related sequences. Again, the gene and gene-related sequence also further divided into coding DNA and non-coding DNA. How many percentage of coding DNA means? 10% only the coding the amino acids or particular diseases, particular like thing. Remaining 90% of non-coding, we cannot be able to identify it. Non-coding means they cannot represent any protein molecule, any amino acids. Likewise. Okay. From these part, they may be divided into three types of genes: pseudogenes, gene fragments, intron, untranslated sequences, etc. These are all are considered the nuclear genome. For example, any one of the plant or animal, the nuclear genome present in it, the nuclear genome is divided like this. And mitochondria, this is also extra chromosome. It's mitochondria, inside there is a genome is there. That genome is also divided into, see that 16.6 kp globase, 37 genes only present here. Here 30,000 genes, human genomes. Okay. See here what happened. 37 genes, 2 RNA genes, 22 tRNA genes, 13 polypeptide encoding genes. Calculate it. 2, 24, 37. Okay. Tally, isn't it? Is it correctly tally out? Here. 37, 2, 22, 13 polypeptide. Encoding gene. This one is also tRNA gene. This one is called as rRNA genes. Okay. This is the what? The extra genetic movement. So, okay. It's divided into 2 parts. One is unique or low copy number. Low copy number. High repetitive number. So 80% will be a low copy number, and the 20% will be moderate to high repetitive. This is also further divided to the moderate to high repetitive, divided into tentamly repeated or cluster repeats, interspread repeats. No need this one. So this part is very, very important. Nuclear genome and this part only. So no need this part. Okay. Human genome, nuclear genome, and mitochondria, otherwise called as extra chromosome. Nuclear genome contain how much? How they are divided. Okay. Extragenic DNA come to here in this part. Genome and gene related sequence. This is the very, very important. How many percentage of coding genes or DNA present in the human genome? You have to calculate. See here, my dear students. This is the 30,000, isn't it? 30,000, 30% is how much you have to calculate. From this one, coding gene is 10%. You have to calculate from the 30% to 10%. Which value comes from this part, 30%. From this one to 10%, you have to calculate. So the remaining thing, how much? This is also a very, very important. Sometimes they may ask the question, direct question. Sometimes they may be a calculative manner, mathematical aspect, they will ask the questions. This is 30,000 means 30% how much? You have to calculate. I think 9,000 like that. Isn't it? From this 10%, how much? 9,000 means 900. Okay. Likewise, you have to calculate. Just check it and then come to. Okay. Go to the next one. In hallmark, their proposal was very, very important. Base pairing between the 2 strands of the polynucleotide chain. This was based on the observation of Ergim Charkov's. Okay. Charkov observed was that for the double-stranded DNA, the ratio between the adenine and thymine, already we discussed, these two are equal, and the guanine and cytosine are constant and are equal 1. Okay. So this is the Charkov rules. One adenine, one purine molecule combined with the another pyrimidine. These two are joined to the double bond. This number of adenine is also equal to the thymine. Okay. This is very, very important. This is the graph Charkov rules. See that. Salient features of the double helical structure of the DNA. What are the salient features present in it? Already discussed it. Again and again, it will repeat it in your book. Okay. DNA consists of 2 polynucleotide chain. The backbone is constituted of the sugar phosphate and also a bases. The 2 chains of the DNA run in antiparallel fashion with 5' to 3' and 5' to 3' polarity in one and 3' to 5'. 3' to 5' polarity in the other chain. Okay. One complete tan is called as pitch or gyri. One complete rotation is called as what is that? Pitch or gyri. Each tan of the double helix or the pitch of the helix is 3.4 nanometer. That is equal to 34 Armstrong. Okay. Go to the next one. It has roughly 1010 base pair in each tan. Each tan. 10 base pair, particularly 10 base pair. Double time repeated here. Okay. The distance between the 2 adjacent base pair is approximately equal to 0.34 nanometer. 3.4 Armstrong. So 3.4 nanometer. The diameter of the double helicals is 2 nanometer. That is equal to 20 Armstrong. The distance between the two base pair present in the opposite strand is called as 1 is 0.1 nanometer. That is equal to 11 Armstrong. Okay. So this is very, very important one, my dear students. Don't forget. Okay. See this one. The sugar and phosphate form the backbone of the helix. Already we know. While bases are aligned towards the axis. The bases are hydrogen bonded, double hydrogen bonded, which each other based on the Chargaff rules. Already told you, adenine, thymine, there is a double bond. Guanine, cytosine, triple bond. That is given here. Why they are giving the 2H? Adenine, thymine, 2 hydrogen bond. Cytosine, guanine, 3 hydrogen bond. Already we studied in the structural aspects. Okay. The plan of one base pair stacks over the others in the double helix. This in addition to hydrogen bonds confers stability to the helical structure. Each one it has a one stack above the others, one above others, like this, arranged with the help of the, which one hydrogen bond. Then only they maintain the structure of helical nature. Otherwise, what happening? It may be differ or collapsed. Okay. The 10 of the helix form a large grooves called major grooves, which is 2.2 nanometer, otherwise 22 Armstrong. And the minor groove, which is about 1.2 nanometer, that is equal to 12 Armstrong. Major groove, how many Armstrong is there? Sometimes they ask the question. Minor grooves, how much is there? So the difference is 10 nanometer. See that 1.2, here 2.2. Major is 2.2. Minor is 1.2. These grooves are responsible for the binding with proteins and enzymes to regulate the functions of DNA and provide stability to the structure of the DNA molecule. This is also very, very important. See the structure, you can easily identify. See that? This is the major grooves. This is the minor grooves. Okay? Minor grooves. These are the bases. See the distance between one nitrogen base to another nitrogen base. This is the total, 10. Hydrogen bond between the base pairs. Here, this one is a blue color is mentioned as adenine, means the red color is thymine. In between, there is a black color. So this is a bond. That's a hydrogen bond between the bases. Suppose this bond is not there. What happened? Each strand it will be separated. Okay. So this bond is very, very important. So when it heated, what happened? Up to 90 degrees Celsius, the two strands are separated. When you are cooling, it will be reunited. See the distance between those two, 20 Armstrong. Okay, so this is already, you know about all those things. See the forms of DNA. This is very, very important, my dear. According to their structure, according to the number of base pairs. See here, how many numbers of base pair is there? Different numbers, 9, 10, 8, 9, 10, 11, and 12. So these are the 5 types of the DNA molecule present in it. Our standard, this double standard helical structure only in the B format. See that B form means right-handed helical structure. Watson-Crick model. How many base pair present in it? 10 base pair. The sugar and phosphate backbone is very smooth model. The axis is rises is 3.37 Armstrong. Length of the one pitch is 34 Armstrong. Already we studied. The diameter is 23. These are the things. These are the characters. So these characters already present in the which one DNA model, Watson-Crick model, otherwise called as B form, right-handed. Already we studied, isn't it? But others also present in it, but in between there is a variation. Here the 10 base pair is present in it, no? But another model, A model, 11 base pairs is there in between the each and every twist. Some of them are 9, some of them are 8, some of them are having 12. See here, A, B, and C, and D. These are all are right-handed only, jet only left-handed. Note it down, my dear. This is very, very important. This is also, this question also asked by the NEET. Which one is jet model? Left-handed model, also called as jet DNA, D DNA, B DNA, C DNA. Likewise asked. And one more is that according to the nitrogen bases, which one is the lowest base pair? 8-1. So D DNA is the lowest. Which one is the highest one? 12. So jet DNA is the highest one. Double stranded helical Watson, which one? Number of base pair is 10. See here, all are smooth in nature. The sp backbone. Okay. All are smooth. This one is called a zigzag. That's why it is called a jet DNA. Zigzag DNA, jet DNA. Left-hander. 12 base pair is there. Axial arises 2.56 Armstrong, 3.37 Armstrong, 3.32 Armstrongs, 3.03 Armstrong, 3.8 Armstrong. First one is 2.56. Others are 3 and more. Length of the one pitch. This is very, very important. Just imagine this is 34 Armstrong only. Isn't it? 34 Armstrong. So remaining things are, what is that? Here, 28.16 Armstrong. Because there are, here, see that. The distance will be very less. See, this distance is very 23 Armstrong diameter. Length is height. Length is, what is that? In between the diameter is very long compared to this one. Compared to this one. See, 19, 18, 20 only is there. But here the 23 Armstrongs. The diameter is very high compared to others. So the axle will be, what is that? 2.56 Armstrong only. Okay, see here, 3.37, 3.32, 3.3. So already discussed these things. 28.16 Armstrong. Length of the one pitch is 26 only. Okay, this is 34. This is 31, 24, 24 Armstrong. And 46.6 Armstrong. All are high in this category. See here, except this one. Because it is very less in nature. 18 Armstrong is very less compared to the other things. But others are very high compared to others. See here, 3.8, 3.37 only here. 45.6 is here. This one is very less. Which one? Diameter is short. So this is 19A, this is 18A, this is 20A, 23. These are the very, very important. See here, there is no diameter. Okay. So sometimes they may ask the questions. In ddNA, the diameter will be 0. I have to mention. Okay. See the structures also. B, Z, A, and C. This is handwritten notes. Okay. So right-hander, left-hander, right-hander, right. All are right except Z only. So already we discussed. 34 arms, 46, 25, 30 Armstrong, 24 Armstrong. Diameter 20 Armstrong, already in our about 18, 26, 19, there is no. Stable, psychologically active forms. Unstable, 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 unstable. B form only stable compound. Sometimes they ask, which one of the DNA molecules is stable other than ZDA, like that. Match the following. Okay, my dear. So this is the structure. See that? This is the B form structure. See that? B form structures. Sorry, B DNA. This is A form, this is B form. This is the DNA model. Okay, B form structure. A form is, this one is A form. This one is Z form. See that? This is the levo rotary, left rotators. So this one is right rotation. This one is also right. This one left. The structure will be like this. Okay. So this is different types of the DNA molecules, right rotary or left rotary. Z only left. All are right. Don't forget, my dear. Okay, this is another part. Same things is there. Okay. See here. No need this one. This one is also. This handwritten notes. Okay, taken from the net. And sometimes satellite DNA also present in it. The size of the repeated unit. How many molecules present in it? Alpha, beta, mini satellite, micro satellite. Likewise, is that these are the chromosomes. Okay. No need this one. We'll see the things. Next one. And next we will pass on to the next one is linear double standard DNA. Already we studied. What is that? Double standard helical structure. But now we are going to study which one, linear double standard DNA molecule. Where it present? Linear means chain, continuous. There is no any rotation. There is no any twisted format. Only linear aspects only. It was identified in the one of the microorganism, particularly one of the mycoplasma. Already we studied mycoplasma. The name of the mycoplasma is called as PPLO. Note it down, my dear. Linear double-stranded DNA was observed in the PPLO. What is meant by PPLO? See here, pneumonia-like organisms. Okay, pneumonia-like organism. PPLO expansion. Which one you know? Pneumonia-like organism. Already we studied this one. Okay, pneumonia. The micro welfare, isn't it? Microorganism used for the biological aspects. That things, pneumonia. It's one of the mycoplasma. What is the meaning of mycoplasma? You know very well. Will you please tell me what is my mycoplasma, my dear students? Mycoplasma, they do not have the cell wall structure. Okay, they only produce diseases and harmful to the organism. So they are all are called as what is a pathogen. Mycoplasma is a pathogen. They do not have the cell wall. They are mostly lived in the anaerobic conditions. They only lived in the anaerobic conditions. Absence of oxygen only they can survive. And they produce diseases, plant as well as the animals, particularly pneumonia in human beings, respiratory disorder, already we studied. Okay, and also, and produce the blue baby. That things also already we studied, isn't it? Don't forget, my dears. But just for the information only, in this category, you must note it down. Linear double-stranded DNA molecule. The full form of a PPLO is pneumonia-like organisms, and the term used to describe mycoplasmas. Okay. The PPLO already told you, mycoplasma general characteristic features. They do not have the cell wall. Okay. They are fully harmful, but they only live in the absence of oxygen, anaerobic conditions only. Okay. They are all pathogen. They produce the plant as well as the animals diseases, particularly lungs in human beings. PPLO is the smallest cell or organisms with a size between 0.1 and 0.3 millimeter. Okay, the size of the PPLO will be very less in nature. Mycoplasma has included species discovered from pleural fluid, note it down, my dear, of cattle suffering from the pneumonia. Some of them are pleural fluid present in, pleural lungs. Okay, pleural layer is there. In between the pleural layer, there is a fluid is there. The fluid, what happened inside the lungs? Will you please, Dhanalakshmi, idea? Pleural fluid is, what is the main use of the pleural fluid? Or Ajay, do you know, Pa? Ajay? Hello? Okay. Pleural fluid of the cattle suffering from pneumonia. Okay. See here. The mycoplasmas are smallest living organism on the earth with no cell wall at all. Even in the lack of oxygen, this organism can survive. Many of these mycoplasmas are pathogen to the animals and plants. This is very, very important, already told you, isn't it? And molecules are another name for the mycoplasmal bacteria. Ajay, do you understand? Okay. See here. So this is the mycoplasma structures already in our PPLO in pneumonia-like organism. The mycoplasma, they do not have the cell wall structure. At the same time, it is outermost covering is that is a lipoprotein membrane. That's a soluble protein, and also DNA molecule and soluble RNA also present in it. That is very, very important. Ribosome also present. Okay. What ribosome present in lower 70S ribosomes. Okay. So they are the simplest and thinnest free living prokaryote on the earth. Mycoplasmal bacteria have been found in the pleural cavity of the cattle suffering from the pneumonia. These organisms are often called MLO, mycoplasma-like organisms. or PPLO, pleuropneumonia-like organisms. Okay, so this is very, very important, my dear students. MLO, mycoplasma-like organism. Another one is called PPLO. It's also pleuropneumonia-like organism, respectively. See that PPLO, full form in biology, PPLO, pleuropneumonia-like organisms, also known as mycoplasmas. Smallest organism, lack of cell wall, single-celled, survive without oxygen. That is called as anaerobic condition. They can easily survive, but they are all what is that? Pathogenicity. Okay, so go to the next one. Palindromic sequence. What is the meaning of palindromic sequence? A palindromic sequence is a sequence made up of nucleic acid with the double helix of the DNA and RNA. That is the same when read from 5' to 3' on one strand and 5' to 3' on the other strands, complementary strand. It is also called as, what is that? Palindrome or an inverted reverse sequence. What is by palindromic sequence, my dear students? Ajay. Dhanalakshmi?
Sir?
What is my palindromic sequence?
Sir, if we read by the both the side, same spelling will come, sir.
Okay, very good. Very good. Exactly correct. What is my PPLO? What is my PPLO?
Don't know, sir.
You don't know what?
No, sir.
Just now I taught to you. See that? Or you see that board?
Pleuropneumonia like organisms.
Yes, yes. Okay. See the pairing of nucleotide with the DNA, double helix is complementary, which consists of the adenine pairing with either thymidine, DNA. Okay. So uracil in RNA. While the cytosine pair with the guanine, already you know about. So if a sequence of palindromic, the nucleotide sequence of one strand would be the same as the reverse complementary of the other strand. That one is Dhanalakshmi already told you. Okay, good, well done. And examples of palindromic sequences is 5' started from the GGATCC 3', which has a complementary strand. Which one you know? CCTAGC 5'. See, after this one, C here, T is also there, and A is also there, G and G. Likewise, 5' to 3', 3' to 5', both are, what is that? Complementary to each other, same. Okay. Given examples of palindromic gene, my dear. Dhanalakshmi. Give an example other than this one. Ajay. Okay. So this is the sequence where the restriction endonucleases and PAMH1 binds to and cleave at a specific cleavage size. Okay. When we are going to cut any one of the portions from the particular nucleotide, we can use it the restriction endonuclease and the BAMH1. It can, what is a cleave at a specific site. Okay. That is called as restriction endonucleases. When the complementary strand is read backward, the sequence is 5' GGATCC 3', which is identical to the first one, making it a palindromic sequence. Okay. So another restriction enzyme called as E. coli. Note it down, my dear students. E. coli mostly found in where? In the E. coli. That's why this enzyme is called as, what is that? E. coli 1. E. coli 1. Dhanalakshmi, are you here? Ajay? Okay. So another restriction enzyme is called as E. coli. Recognize and cleavage the following palindromic sequences. Okay. See the 5 dash to 3 dash. See that G, A, T, T, C. So G complement with C, A complement with T, A is also T, T is also A, T is also joined with the A, and C is G. See the 5 dash to 3 dash and 5 dash started from the G. In this part, 5 dash started from the G. Likewise, this is the pair of the sequences, each one. Okay, that is called as polyandrome. Likewise, the Malayalam is also there. Malayalam. Okay, this is one of the word. It is also examples of which one? It is also an example of which one? Polyandrome. When you are reading the Malayalam, okay, the opposite is also the Malayalam. It comes. This is the examples, the best examples of which one? Polyandrome genome. Okay, it's not like that. It is similar to that of the which one? 5 dash to 3 dash sequences, as well as the 3 dash to 5 dash complementary gene sequences. Okay, this is very, very important, my dear students. Don't forget this one. So denaturation and renaturations. Already we studied these things. Separation of two strands of the DNA from each other due to the breakage of hydrogen bond only. When it is exposed to the high temperature, acid, alkali is called as denaturation, or what is that? Melting. As a result, you will get which one? Two single strand separately, isn't it? And reassociation of the separated DNA by hydrogen bond formation is called as renaturation, otherwise called as annealing. Don't forget, my dears, annealing. DNA with more AT has a low melting areas. Don't forget, my dear, DNA with more AT has low melting areas and denaturation more easily. DNA with more GC, that AT, high melting area. Why? Ajay? Why? The adenosine area will be very low melting areas. Suppose if it is a guanine and cytosine area, it will be high melting area. Means, you know well about adenosines combine with the molecules of thymine with 2 hydrogen bond only. But at the same time, guanosine or guanine join with the triple bond. So the one more hydrogen bonds is present where the guanine and cytosine in between. So the breakage will be very high compared to the adenine and thymine. This is also a important one. See, C value. What is the meaning of C value? The total amount of DNA per genome is called as, what is that? C value. The amount of DNA is expressed in the picogram. The amount of DNA is expressed in the picogram. 1 picogram, that is equal to 10 power of minus 12. That is called as 10 power of minus 12. You have to put the upper grams. 1 picogram, that is equal to, sometimes they ask the questions. Okay. DNA, amount of the DNA will be expressed in the term is called as a picogram. The 1 picogram, that is equal to 10 to the power of minus 12 grams. Don't forget, my dear. Okay. This is very, very important. Okay. Go to the next one. What are the main functions of the DNA molecules? Already you know well about. DNA molecules act as hereditary informations. It transfer the one generation to another generation. This is very, very important. Second one is the DNA molecules variations that occurs due to the crossing over. Due to the crossing over only, what happen? The variation will be occurs. Otherwise, it will not happen. DNA variation, when it occurs crossing overs. At the time of meiosis only, mitosis did not happen, isn't it? Because there is no crossing over takes place in the mitosis. Okay. In meiosis, the two gametes are united together. The crossing over takes place in the meiosis only. At the same time, the mutation is also. What is the meaning of mutation? Mutation is nothing but the sudden inheritable variation due to the time of meiosis. What is a mutation? Sudden inheritable changes takes place in the chromosome or nucleotide or base pair. Okay. That is very, very important. And fourth one is autocatalytic functions or DNA replication is called as, what is that? DNA to DNA. That is called as autocatalytic functions. This is called as replication process. DNA produce DNA. That condition is called as, what is that? DNA replications. And then the heterocatalytic function. What is the meaning of heterocatalytic? Other than the DNA molecule, they will not produce the DNA. Other than the DNA means DNA can produce RNA molecules. RNA can produce which one? No, protein molecules. DNA to RNA transcription. With the help of the DNA and RNA, the protein molecule synthesis takes place. That condition is called as translation. And also hormone synthesis also takes place. These are the heterocatalytic functions done by DNA molecules. And DNA acts as a control of the metabolism, growth, and differentiation. DNA is very, very important for the overall control in every cells, particularly protein synthesis, enzyme synthesis, hormone synthesis, other components, metabolics, growth and differentiations. The last one is called as, what is that? DNA fingerprinting technique. What is the main use of the fingerprinting technique? Well about it is very, very essential for the, what is that? Crime reports. Any other murder or any other malpractice done by the particular culprit, they can easily identify it with the help of the DNA fingerprinting. So these are the important characteristics of the DNA. So I think the first lesson, first chapter is finished. First part only finished it. So I will summarize all those things until wait for some minutes. Today, what we have discussed, what is by DNA molecules, isn't it? What is by DNA? Double helical structures. It is also, about there are two polynucleotide antiparallel to each other with the help of the hydrogen bonds. The backbone is sugar and phosphates. Okay. So this is the structures. Already we saw it. And next one is, what is that? Right-handed helical structure is that the base pair, 10 base pair is there each and every rotations, and also 34 Armstrong is there. The extranuclear presence in the other than the nucleus is called as, what is the extranuclear component. It's mostly found in two components. Is there examples? One is called as mitochondria, another one is called as chloroplast. Okay, these are the structures. Already we saw it. The extra chromosomal body is also circular nature. Okay, so another one is called as, what is that? Packaging of DNA. The total number of base pair is 6.6 into 10 to the power of 9. Distance between two base pair is called as 0.34 nanometer. Therefore, length of the DNA, you have to multiply these two, you will get it the 2.2 meters. Okay. So this is very, very important. And size is also vary, depends upon the length and nucleotide. In bacteriophage 174 has a 55,386 nucleotides. Bacteriophage lambda 48,502 base pairs. E. coli, it has a 4.6 10 to the power of 6 base pairs. And then human genome. See that? And DNA is an acidic substance. Already this was discovered by the name of the scientist called as Frederick Meisner in the year 1869. He named acetine, which one nuclein. In human genomes, haploid nature, note it down, my dear student. This is very, very important. 3.3 into 10 to the power of 9 base pair. This is very, very important. The structure of double-handed spherical structure was discovered by name of the scientist Watson Crick and Francis, based on the which one X-ray diffractions by Maverick Wilson and Rosalind Franklin. Okay, what is by XRD? X-ray diffractions. This is the X-ray diffraction is one of the method and used for the crystallography and identification of any solid substances. Okay, this is the methods. Already we saw it. This one is the technique, principles maximum used. In between there is the X-rays comes out from the instruments, and there is a center, there is a solid material and scatter different diffracted rays in the films, you will get it. And as a result, we can easily identify what are the component present in it. Okay, this is another fault. This is the structure. We can see it in the film. So, human genome is also divided in two parts already in our nuclear genome and mitochondrial genome. Nuclear genome is also divided into gene and also extra genes. Genes also coding and non-coding. Which one is coding? Protein, amino acids, hormones, like that. Some of them are 90% of non-coding substances present in it. Okay, coding substance only 10% only present in it. Okay, this thing is also very, very important. Don't forget, my dear students. Okay, this is a very important questions. And Charkov rules also already in our adenine always combined with the molecules of thymine, cytosine combined with the molecules of what is a guanine with the respective bond. Okay, and also the number of adenine molecules equal to number of thymine, also number of guanine molecules equal to number of cytosines. Okay, this is also very, very important ones. And then salient features already we discussed, same as it is in the previous one. How many base pair is there? How many Armstrong is there? Okay, in between the diameter, how much is there? How many bond is also present in it? And also the major groove and minor grooves also, how many Armstrong? 2.2 nanometer in the major grooves, and minor grooves 1.2 nanometer is there. And so types of DNA molecules. Okay, forms of DNA A and B and C and D and D. Jet DNA. So jet only the difference other than compared to this one. Okay, B DNA is the we studied one. All are right-handed. These are the base pairs also present in smooth. The phosphate as well as the sugar backbone is a smooth, and which one is the zigzag? That way it's called as a jet DNA. And length of the DNA. These are the things. Diameter also it vary depends upon the forms. Okay, so already we saw it. This one is also, and these are the structures already we saw it. And then DNA is also another one, standard linear double stranded DNA molecules we studied in the PPL work. Okay, this is pleuropneumonia like organisms. It's a mycoplasma, smallest one, 0.1 to 0.3 nanometer, mostly found in the cattle suffering from the pleuropneumonia. So things. See that the mycoplasma are smallest and living organism on the earth. There's no cell wall. This is very, very important. Lack of oxygen, they will be survive. They only produce the disease only. That's why it is a harmful effect only present in the animals and plants also. Some molecules. It's another name for the mycoplasma bacteria. See the structures already in our about these things also already we saw it. Okay, and then palindromic sequences also similar to that of the Malayalams. Okay, when you are reading the 5' to 3' and 3' to 5', both are same. Okay, so example is this one is called as already we discussed GGAT-CC. You have to start from the GGAT-CC. It's a complementary to each other. Likewise, Malayalam, palindromic also Malayalam. When you are reading the Malayalam, opposite side reading also same Malayalam. Okay, and also duration and denaturation. When you are heating, what happens? Double banded is also broken, and also annealing means cooling process also. So this is also very, very 1 picogram that is equal to 10 power of minus 12, 10 power of minus 2 that is equal to 1 picogram. That is also depends upon the amount of DNA is expressed in picogram. So number of amount, how much amount will be present in the living organism. The amount of the DNA is expressed in the picogram unit. So 1 picogram, sometimes they may ask the question, 1 picogram that is equal to 10 to the power of minus 12 grams. Don't forget, my dears. And DNA functions already know hereditary information, and mostly crossing over takes place in meiosis. Then it the variations occurs, mutations also. But it's by replication, DNA to DNA produce replications, and heterocatalytic function. What are the heterocatalytic function? Means DNA to RNA, proteins, hormone synthesis, and also metabolisms and different metabolic hormones, enzymes, and also other metabolic functions also done by the DNA molecules. And last one is DNA fingerprints. They can easily mostly used for the culprits identifications. So I think the first part will be finished. We will see the remaining on the next classes, my dear students. Thank you so much for the participation. Thank you, ma'am. Thank you so much. Thank you, Ajay. Thank you, ma'am. Thank you.